Now that we've looked at the anatomical structure of a neuron, let's now turn our attention to first some basic functional anatomy of neurons. So we're going to want to talk about polarity or membrane potential. Specifically, we'll focus on resting potential at first. And then we want to look at these structures called gated ion channels and sodium-potassium pumps. Then we'll look at basic states of a neuron. So we'll talk about depolarization, hyperpolarization, action potentials, refractory periods, and spiking frequencies. So neurons have what's called a membrane potential or polarity, and it's just the net difference in electrical charge between the inside and the outside of the neuron, measured inside relative to outside. Resting potential is the normal polarity, the homeostasis, and it's measured by that difference inside relative to outside. For most neurons, it's approximately negative 65 millivolts, but it ranges amongst neurons from negative 40 millivolts to negative 80 millivolts. Resting potential is maintained by sodium-potassium pumps and by non-gated channels, which act to keep the internal level of potassium high relative to sodium. Now, since neurons have in their body, in their inside, negatively charged ions, anions, mainly organic acids and proteins, the net charge inside and a neuron is negative. So here's a drawing of the relative resting ion distribution around non-gated channels and a sodium-potassium pump. So Na is sodium, K is potassium, Cl is chloride, and A is those large negatively charged anions or ions in the membrane proteins. So we can see concentration of potassium and negatively charged anions inside. Outside, we have some chloride, and sodium is more concentrated with some potassium out there. In resting potential, there are approximately 20 times more potassium ions inside a neuron and approximately 9 times as much sodium outside the neuron. Now, this acts to create what are called electrochemical concentration gradients for sodium going in and potassium going out. And that's going to drive the function of our neuron, that action potential. Now you might wonder, how is it that influxes and effluxes of sodium and potassium are going to change things, since, well, they differ in size, as you can see here. They both have a positive charge of 1. Right? Well, there are these concentration gradients. Right? There are electrical concentration gradients where positive tends to flow to negative, negative tends to flow to positive. And there are chemical gradients where the concentration is high. Diffusion wants to push those elements to where the concentration is low. So both passively and actively, there's a transport of sodium out and potassium in to our neuron. But you might wonder, well, how do sodium and potassium efflux and influx change membrane potential, given they're both positively charged? Well, the way that this works is that changes in levels of sodium and potassium change membrane potential because they occur at different times and in different directions and really at different rates. So here's a drawing depicting a non-gated potassium channel at the atomic level. So you can see there's a collection of protein structures here. There's an opening here, and that would be where our potassium can flow into our neuron. Now, potential can change in a way that's excitatory, called depolarization, when the difference between the inside relative to the outside changes so that I have a decreased potential, say from negative 65 millivolts to negative 60 millivolts. We can also have inhibitory or hyperpolarization. And that's where changes in the membrane potential measured from inside to outside are such that there's a greater polarity, say from negative 65 to negative 80 millivolts. We can also see neurons exhibiting what's called an action potential down their axon. So when a change in potential at the axon hillock, that structure where the axon leaves the soma or cell body, causes a much greater change to occur, usually from negative 65 volts to close to positive 40 millivolts in the polarity of the axon at that spot, that triggers a transitory event that then travels down the axon. Now, one thing I should point out, as I noted before, the resting potential of particular neurons is somewhat variable, and the change in potential as a result of an action potential. Positive 40 millivolts is a common number that you see, but they can vary in their how high and how low they get during an action potential as well. These are just the most common numbers that you see. Now, after there is a spike, an action potential that travels down the axon, that area of the axon enters into what's called a refractory period to recover. And during the refractory period, there cannot be a spike traveling down that part of the axon again. And this means there's a limit to the number of spikes or action potentials that a neuron can transmit. And it's sometimes called spiking frequency. The number of spikes possible during a second is limited to about 500 a second, though neurons generally don't come close to that rate. Periphery neurons tend to have a limit of 20 spikes or so a second. Most brain neurons have limits between 200 and 300 spikes per second. And so there are upper bounds to how many times a neuron can spike. In our next module, then, we want to take a look at the process of an action potential from beginning to end. And then we'll end this lecture by looking at different kinds of neurons.